I'm from IT Kanpur. Yeah. So you're doing the. Okay, I know the name and faculty mapping. So you're doing the technical stabilization, right? So yeah, you will be doing the talk and yeah. So it will be on Tuesday. So you might have seen the schedule, right? Yes. It's 5 minutes thing, and then of course you are going to do a. And I think there is another student. He's from. He's working with Prasad and Kabir.
Oh, right. Yeah.
Yes.
Yeah.
So 6. So it will be 5 minutes now. What you have to do, you have to prepare it in a format where we don't need to change the laptops. Okay. So it has to be in a more unified way, so that we do not lose time in doing change. So we'll try to make it like 4 to 4.5 plus 30 seconds or 1 minute, so that there will be option for one quick question. Okay. So it will not like you just speak and then you disappear. So at least the important question can be answered. Okay. So you have to be strict. So don't go for very long introduction where you lose 3.5 minutes and then you say, oh, I haven't even started. So have you guys given that work before? Yes. Okay. So it's a good. So for you, maybe not, but for them, then you have given. Okay, fine. So this is very common practice nowadays, and it's a good training. Okay. So is it ready? Yeah. So guys, any questions? So Matthias is ready to. And then I think you will start the lectures. Right?
Yeah. Can I use that? What was the trick? Should just second screen. Actually. Not yet. Yeah. I'm gonna wait that this guy pops up. I don't know. Is it? Oh.
It is not coming on the screen. Oh, there is.
Here there is. Here there is. Let me actually check. Is there power here? Yeah, there is. Okay, there is. Should you see something here?
Yeah.
So maybe the cable or is this? Oh, yeah. Wait. Okay.
Yeah.
And
okay. So there are no such impacts of the base community without the results. Okay.
can you actually, let's see. Do you see it there? Okay, good. Okay, so today what I want to do, guys, because I already talked about a little bit the coherent structures in the chick embryo, which I'm gonna refresh for those who were not here yesterday. But then, I want you to understand, or address several of you were asking, okay, but what actually causes these coherent structures, right? Can you understand what forces generates the flows and are such that you generate some particular coherent structures? And today I'm going to actually discuss this problem in the context of the chick embryo, and we're going to see that actually to describe this system, you have to use the PDEs that are similar to what Anupam was discussing, but they're the active nematic PDEs, which is also Anupam, I think, flashed it yesterday. And then we're also going to go, if you know how to basically construct or predict these flows, then the next level would be, can we actually control them? Okay, so that's what you are going to do. So this work is a long-standing collaboration with the group of Kees Beher at the University of Dundee. Kees is a developmental biologist that did all the experiments. Guillermo was a student with Kees and now is a postdoc in Cambridge with Ben Steventon. They also did other experiments. And Alex is a PhD student in the group. Okay, so what we are going to do today is... So this is our system, okay? So we discussed yesterday. So for us today, the living system or the flow is just a layer of cells that is the chick embryo, the beginning of development. You can see it from opening an egg. And it's very important to understand this stage of development because two reasons. The first one is that the chick system resembles human gastrulation, which is this very early stage of development. It's called gastrulation, and of course you cannot study this process in humans because the cells are inside the body of the mom, so it's really inaccessible. So biologists, what they do, they consider model systems or animal systems that are the closest, that they share basically the similarities with the human similar developmental stage, okay? And the chick is one of the vertebrates that shares this process the most closer to human. So basically this is how you want to try to understand what happens in human. Why this is important, again, because this is when you establish your main body axis and bilateral symmetry, and this is also when the first germ layers are starting to form. So basically if you mess up this stage of development, it's very, you're going to have very likely irreversible consequences. Okay, that's basically the biological relevance of looking at this system. And the physical relevance is that really this system is basically two-dimensional. You can really image. You put a microscope on top, and therefore it's also interesting to understand how basically can we learn new mechanisms of living systems looking at this basically in vivo tissue. Okay, so what we did, and if you really want to have it just a little bit of a broader overview, we are looking at this, first 15 or 12 hours of development, when you start from a disk of cells, two-dimensional, to now an elongated, symmetry broken, if you will, make, at least geometrical symmetry broken system, and then if you wait 18 days, basically you're gonna get the full embryo, the full animal, basically. Okay, so. Well, what I'm going to do, basically, I'm going to first go through what I showed quickly yesterday. So how can we quantify these multicellular flows using coherent structures? And then we're going to understand how to predict these flows and eventually how to control them, okay? And, as usual, stop me anytime. I mean, I don't have, usually I try always to calibrate slides in a way that there is plenty of time for questions, okay? Okay, so general question is, we have this tissue flow. This is now very clear, so I'm going to go relatively fast. If you start looking at this basically velocity field, regardless of what it is that is moving, you can either have an Eulerian description of the flow, which is basically, you know, for instance, you can look at, I don't know, velocity fields and streamlines. If you look at velocity field and streamlines, you're going to get some sort of, you know, plots like this, okay? However, the problem with looking at these plots, especially in this context, but in general, is that the geometry of these plots are basically frame dependent. That means basically, for instance, that think about this embryo is actually it's happening. At some point, the embryo starts drifting to the right. Okay, just because maybe one edge here detached from the substrate on which is, you know, is originally attached, and then everything drifts to the right. So just that thing that can very likely happen is going to change completely the geometry of this flow. And in fact, you can see here that everything is sort of translating to the right. Okay, so now another embryo doesn't do that. Another embryo may be translating north. There could be relative rotation with respect to each other. So if you want to assess the material response of this system, you don't want to use these techniques because they depend on the frame. Instead, you think about it, the embryo develops in a way that really doesn't care about what kind of coordinate system am I picking to describe the motion of its cell. That's something that shouldn't really affect any conclusion on where things converge, diverge, or stretch, okay? And this is basically the notion of frame invariance, which is that, you know, the material response of a deforming continuum, could be a living continuum, but also could be a passive continuum, should be independent of the observer of the reference frame. So the velocity fields, also the vorticity, the Q criterion, or the helicity, and all this kind of stuff are all frame dependent. So we want to look for quantities that are frame invariant to describe the material response of a flow. So one option to do that is basically look at Lagrangian trajectories. So you start saying, okay, now I want to, instead of looking at fixed Eulerian position, I'm going to go to the Lagrangian coordinates, where I will remember if I start here, where I will go after certain time t. Now this must be very clear, and then if you take now the gradient of this flow map or trajectory map, then you can also understand how an initial spherical neighborhood is going to deform. This is what we did in the last few days. Okay, if you want to visualize this tensor a little bit intuitively or naively, you can just think of looking at this embryo and deforming a Lagrangian grid on top of it. So now the beautiful thing is that every square is labeling a patch of tissue, so you can follow where it goes, you can follow how it deforms, and that's basically the same patch of tissue. You just label every patch of tissue, right? Okay, so we've seen basically the deformation gradient is what popped up yesterday. And, well, I mean, I didn't tell you yesterday, but, yeah?
Just, so these Lagrangian agents are cells or tissue?
They could be any, right? So it depends. In this case, the dynamics is given via velocity field. That means so you basically do images, they do images, they stay in cell membranes, so you have some intensity field of cell membranes, and then with this snapshot of images you do PIV, which you reconstruct some tissue scale velocity. So it's pretty high resolution, but it's not cell scale resolution, okay?
Higher than cell scale.
Higher than cell resolution, but we also have trajectories that cell resolution doesn't really change. It's really, the resolution of the data is going to tell you how precise you can be in attractors, repellers, stuff.
But it gives the trap only if there are cells, because it is that the tracer, the fluorescent materials are attached to the membrane only, right? So not around the cell. So around the cell there could be extracellular matrix, and there may be dynamics around that also. But it's just
this is an epithelium, so
okay,
this is an epithelium. But yeah, if you have a mesenchymal situation, then you have to decide what to track. If you want to track the nuclei or
in this case it is fine.
Yeah. Yeah.
So yeah, fine. So in that sense, like we have the information of the flow.
Yeah. You have information of the flow and of the cells, basically. Yeah, they move together with their membrane.
Yes.
Yeah. Okay, so an exercise that you can actually do is that basically, the largest eigenvalue of Cauchy green, the square root of that is the largest singular value of df. It's an exercise that you can ask always as an exercise. If you have a linear relationship which is like that, and, A is a matrix, 2 by 2 in this case, but in general, and you ask maximize, what is the maximization with respect to x of norm of ax divided by x? Do you know what's the answer? So you take a matrix, multiply by a vector, you get another vector, and I'm asking, okay, you can, the matrix is given, and you can pick the vector. What's gonna be the vector that maximizes the norm of ax, which is the output vector divided by the initial vector? It's not the eigenvalue. It's the eigenvalue only if A has some particular property, symmetric, etc. But if A is a general matrix, like basically df, the answer is actually the largest singular value of A. Okay, so the largest singular value of A, so if you want to remember zeta t is equal df zeta 0, okay, so if you want to maximize df zeta 0 divided by zeta 0, this is the largest singular values of df, which is also equal to the square root of the largest eigenvalue of C. Okay, it's just anything. So basically, basically you can compute the Lyapunov exponent just computing df. You don't need to do even the Cauchy-Green. Okay, doesn't matter, it's the same. Actually check that it's the same and also check it mathematically. Okay, so basically that's what we did yesterday. So, okay, looking at this matrix is a little bit laborious and not really visually appealing. So I'm gonna look at the largest eigenvalue. And then we defined basically the repellers as basically ridges of this largest eigenvalue. You can basically do the logarithm and rescale by time, but these are just, they will not change the topology of the function because the logarithm is a monotonic function. So the maxima of the f will stay the maximum also of the FTLE, or of lambda 2 will also say the maxima of the FTLE. That's basically what we did. Then we computed it for this system. We basically let it go for increasing time interval, and then you see that this system is going to develop basically two repellers, right? Which we discussed yesterday. I'm going to discuss them briefly in a second after I introduce the attractor. This is also what we did yesterday. So you can compute where things initially far away will converge to in a particular time interval, t0, t. And you can also compute the direction of attraction, which is where things, in which direction things will actually converge. And in this system, we had one attractor. Okay, so basically what we did in this compression is using these Lagrangian coherent structures, which in the context of development, and using basically just attractors and repellers, not elliptic coherent structure, parabolic one, computed specifically with the FTLE, we call this dynamic morphoskeleton. Because with these attractors, repellers, and domain of attraction, which I also introduced yesterday, you can basically sort of compress these spatiotemporal flows of, tissues, okay, or embryos. So basically what we did, we started from raw velocities, right, and we just compressed this dynamical system, if you will, into 3 units. So the repeller number 1 splits all these cells that will not be forming the embryo. The attractor marks the primitive streak, where things will go converge and then will enter into the 3rd dimension to start to let 3-dimensionality start to form, which is how you form your internal organs and our body. And then the repeller 2, we saw that basically was splitting the anterior part and the posterior part of the streak, or i.e. the attractor, by splitting its own domain of attraction. That's basically what we did yesterday. Is this clear? Okay, so now it's beautiful because we have 3 units. So you have basically spatiotemporal system given over 12 hours, and we compressed it into 3 units. Think about it. This is basically what you do when you start looking at, classical dynamical system. What you do, start looking at fixed points, compute stable and unstable manifold, then you could look at limit cycles, etc. So you throw away all the rest and you retain all these robust ingredients, okay? This is basically what we are retaining. Yeah.
So the time scale, like for example, repeller one, so maybe that might be appearing earlier. And do they stay for like throughout the process, or do they disappear towards the end, or how does it work? Like
They stay. So we can see that here, right? So sorry. Hey.
So if you have slides later, we can.
No. We can see basically when yeah, you see here. You see this guy forming and stays. It gets sharper and sharper actually.
The second one.
Yeah. I mean, this is a noisy data set. It really sells. Yeah. No, but let me actually show you. This is a very good point. So now that what I think, because we have a little bit of time, so I don't really have to go fast with these slides. But think about it: if you would start comparing these systems, and you wouldn't have some techniques like that, what would you be comparing? There is infinitely many things you can start comparing. You say, okay, what should I look? Should I look at the integrated velocity over the domain, over time, or the vorticity, or I don't know, some statistics of it, modulo the fact that they're all frame dependent. So that means basically that if just an embryo drifts because one edge is detaching, maybe this is due to the mounting artifact of thing, this is going to affect your metric. Okay, instead, all these guys are frame independent. It's okay, this by construction, they don't depend on which frame you use. And they are basically compressing this information into very robust units. This is, think about it, what would you do if you would have to answer, how do I compare these different systems? Okay, so just one thing, so these objects are the most robust features that you can extract from a dynamical system or a velocity field. And in fact, what you can do with very low resolution microscopy, so basically Guillermo Serrano in Cambridge, in the group of Ben Steventon, developed basically a platform in which you can image 9 or 20 embryos at a time. That means compared to the data set that I showed you here is one embryo at a time, but you can have a much higher resolution, a resolution about a couple of cells size per pixel. So it's a very high resolution. So but you have to be very slow in scanning the embryo, and you can only scan one embryo at a time. So Guillermo instead said, okay, you know what, I'm just gonna give up on resolution, but I want a larger N. So he basically now can image 9, 10, 20 embryo at a time. Okay, and therefore the structures will be less resolved than in the previous case. But the interesting thing is that 9 different embryos, the shape of these repellers may change, but there are going to be always 2 repellers. Okay, sometimes there is a third one appearing. We are going to understand why is that the case, because after basically this body recirculates here, things will start separating, similar to the double gyro stuff that you have. So this is not really an artifact. But the long story short is that these are very robust. So now imagine comparing, 9 velocity field over time. Okay, these guys will always be there. Okay, so this is a very strong features that you want to use to compare, noisy data sets. Okay, so what do we have now? We have basically 2 repellers and 1 attractor for this developmental process. And of course, the same for the attractor. I just didn't show the attractor, okay, for robustness. Okay, so now we want to understand, let's try to understand what are the mechanisms that generate this flow. I mean, to address this question, you really have to interrogate the biologists because they know what's going on, okay? Okay, so what we want to try to do now, we want to try to understand, first of all, can we build a model that reproduces this tissue flow? Okay, so that you just, simulate, like the models that Anupam described us yesterday. So can we build one to simulate or to predict these tissue flows? Can we understand what are the origins of attractors and repellers? What are the forces that are generating those? Okay. Then we say, okay, well, how does actually the embryo changes its shape from a disc into a pear shape? And how does it do it? This means basically you require some distribution of forces that shape your boundary, if you will. And can we actually control these guys? Because if you really know how they form, then the brutal way to understand, if you really know how they form, then you say, okay, can I tweak them? Okay, so in vivo. That's what we are gonna try to do. and stop me anytime. So when you basically look at this system, again, remember this guy is a little disk at the beginning, at the initial time. There are two cell types. So there is basically the embryonic cells. Then there is an extra embryonic ring. These are the cells that will eventually be separated by repeller number 1. So they will not form the embryo. And within the embryo, there is some cells in the posterior that are different. They are called mesoderm. And the mesoderm cells are the engine of these systems. Whenever you have cell moving, it's because mesoderms are driving. The active forces are usually in the mesoderm. Okay, this is basically just a way to explain mesoderm. Of course, genetically they are very different. They are different cell types, so we can go also in that direction. But okay, we don't need it to go that direction for this talk. Okay, okay, so this is at the initial time. At the final time, that's what happens to the embryo. These cells are moving outward because they are crawling on a substrate. The extra embryonic tissue expands. You see there is an expansion here. The embryonic basically transforms into a pear shape, and this mesoderm basically goes to the attractor and internalizes. Strictly speaking, also recruit more mesoderm. You lose these engine cells because initially you're on this plane, then you go to the attractor, you leave the plane, but you recruit more mesoderm via induction on the plane. Okay, that's what's happening, but not important. So remember, 3 cell types: extra embryonic, embryonic, mesoderm. The mesoderm is the engine. Okay, and this mesoderm starts from the posterior at the beginning and then goes to the center line. And there is actually also these embryonic cells. So I'm coloring in the light blue. This is a darker blue, light blue, and white. Okay. Okay. So now, what are basically the active processes that are driving this system? So there is basically active intercalation into the mesoderm. Intercalation just means basically that cells that are initially far, they converge. So therefore, they basically exchange neighbors, and therefore they converge, and then they do a kind of a saddle flow. This is what happens. These cells basically do this, and then these two that were initially in contact, they're not in contact anymore. That's basically, they do it by contracting and modulating tension in their junctions, if you want to understand. So that's what's happening in the mesoderm cells. There is a lot of intercalation. Then there is, in this system, there is a lot of apical constriction. So cells basically will decrease their area. And then eventually here there is going to be an ingression, but in general they will decrease their area. So they will contract their area, and therefore the packing, so basically you're going to have a higher cell number density over time. Yeah.
So that disappearance of the cell is going to start developing.
Yeah.
That's what it is.
Yeah, that's but in general, there is also a reduction of apical area of these cells. So the cells basically from a little cylinder, they will squeeze, they will get longer basically. So then there is the active ingression. So basically at the streak, a lot of cells are actually ingressing. So you see that there is a flow of cell, and then these cells are basically, they keep flowing from the sides, and then here they are going to disappear in the 3rd dimension. That's another process that is driving this system. And then there is these edge cells that are basically just crawling on a substrate. This system is very fascinating because all this piece of tissue is suspended. So there is, this is the extra embryonic blurb. So you have this segment of tissue, is that there is no substrate here. Then there is all the epithelium or the embryonic tissue here. And then of course it's going to be symmetric on the left. And then this little peripheral edge is attached to a substrate, and here cells basically using myosin, they develop protrusion and they pull these cells outside. Okay, that's basically what's happening. And wherever you see this motility or wherever you see this activity is driven by myosin. So myosin is a molecular motor that basically synergizes with actin, binds with actin, and contracts. It's basically what we do in our muscles, basically. Yeah.
So the measurement has been made on myosin level, or is there some other protein like Wnt or FGF is also?
You don't really need it at this level.
I'm learned about in experiments when they did the observations? Like, of course, from tracks you can see and also indirectly. But did they ever comment?
There are other studies on wind measurements and FGF, but those are more for differentiation in this case. Because really myosin drives the system. This was established already in, maybe 10 years ago. Yeah. So basically now, so this is what's happening. Now I'm telling you that wherever you see myosin, there is higher activity. So myosin is required to do this active intercalation because you need myosin to contract these junctions. You need myosin to contract your apical area. Just contracting all these junctions together, or these internal filaments will actually shrink this apical area. So, but to do that, you need myosin. To ingress also, you need myosin because actually you have to shrink it so much that eventually you reduce it to a point and ingress, and you need myosin at this edge to move, okay? And if you basically do the experiments, this is how myosin looks like. So basically, this is at this stage, the embryo, wherever you see cyan, this is the active myosin. So you see there is myosin in the embryo, there is more myosin at the attractor. There is no myosin in this extraembryonic region. This is actin, so it's another basically marker that basically, so myosin and actin will bind together, and if you have active myosin, these binded filaments will contract. Here basically you have only active, no myosin, so this tissue is a passive region. Here instead you have both. Those are basically just superimposed. So whenever you see both, that means that there is both actin and myosin. I mean, it's not good quality, but you would see also the red here. Here there is only actin, non myosin, and then there is this myosin on the ring where cells are actually migrating outward. Okay? This is the only biological slide I have. Now we're gonna, maybe there is a little bit of quantification in the next slide. Questions? Okay, so what we want to do, we want to try to build an active stress, okay? So in this tissue, there are basically three regions, I already told you. So if you zoom here, there is no myosin, okay? So this is more of, we think of this as a passive region, okay? There is no myosin. If you go in the embryo, there is a little bit of myosin, you see that there is cyan, and the myosin is pretty much isotropic, it's not much oriented, because myosin is really like in active nematics. You have alpha, which is the parameter of activity, a strength, which now here is not going to be a scalar, constant, it's going to be a scalar field, because some regions will have more contraction or contractility than other regions, okay? That's basically the scalar field, the myosin scalar field, and then you want to understand nematic order parameter and orientation. If myosin is distributed more on a particular direction, which means basically that, I don't know, if I have cells, say if I have a cell, and myosin is more activated in this direction, so there is going to be myosin in this region, but then you also want to say, hey, look, I mean, there is going to be a preferential direction of myosin in this direction, therefore the contraction is going to happen in this direction. So that's exactly how people model active nematics. Okay, and this is these bars that you see here. The yellow bar, we're actually marking from experiments the alignment or anisotropy or directionality, if you will, of this myosin signal. Okay, and you can see basically that at the attractor there is a lot of myosin. This is why the scalar field is brighter, but there is also a lot of orientation. So cables are aligned, or your myosin is distributed preferentially in the perpendicular direction of the attractor. Okay. So this is basically what you can measure, what they measured, and therefore let's, yeah.
So yellow arrows are just average quantity.
Yeah, they are average quantity. Yeah. So you do basically myosin imaging of this, and then you do Fourier transform, and you consider the mean, the anisotropy, and everything. Okay, so what do we need? We need a scalar field myosin activity. Okay. Then we need a nematic tensor. Are you familiar with the nematic tensor? Should be familiar with the nematic tensor. Basically, this is what's, Anupam described it quickly, but again, this is exactly what I said. So if you are, this is going to tell you the orientation of the nematic director, which in this case is the myosin orientation, and S is the nematic order parameter that tells you that goes to 0 when there is no oriented myosin, or it goes to 1 when all the little cables, basically, if you do an average, let's see, if you do an average, basically, on a control volume, and these are all the cells, right? So then each of them will have a particular preferential orientation of myosin. You do this average, and you're gonna pick this is the average value of these cables. Just do the average, and then S is telling you if S is equal to 0, that means basically that they are all randomly oriented, so there is not even a uniform, a particular, well-defined mean. But if myosin is if S is equal to 1, instead that means that all these basically cables, or unit cables, if you can call them, or the orientation of myosin in each single cell is aligned with the mean. So the mean, all the cells are basically contracting myosin in the same direction. That's what it is.
It is very similar to the way director field was defined, if you remember that again. So yellow symbols are the director field, right?
Yeah. Yeah.
And then from that you can construct the cube field that we have seen.
Yeah. I mean, are you familiar with the active nematics, liquid crystals? Okay. Yeah, that's basically the same tensor. Okay, so these are 3 fields, and therefore now we can just build the active stress. We're gonna say, okay, myosin is going to be a fraction of activated myosin, it's basically a concentration. Alpha is going to be a scalar that maps myosin to stress, okay? And then there is going to be an identity tensor that accounts for the isotropic contribution of forces or stresses. This could be associated with ingression. Remember, ingression, if you have a lot of myosin here, you're just going to contract isotropically, so you're going to generate an isotropic active stress. And instead, if you have the anisotropic one, that's what's going to actually model the active intercalations. Okay, I'm going to actually contract here and go in the other direction. Okay. So, yeah.
From the data, can you differentiate how much isotropic and how much anisotropic?
Yeah, that's exactly that, right? The mean is going to be the mean. You can basically, if you have a scalar field, you can understand what's the average of the scalar field, and then you can also ask, is there a preferential orientation, and how much is distributed in that particular orientation?
Okay, I see. So you do a vector sum and scalar sum, and that will give you the
There is a PRE paper that now I don't remember the authors and the name, but it's in the citation in the paper, in our paper cited, in which you can extract these quantities from the measurements of myosin. Yeah. Okay, so are we on board with this? Okay.
So please stop if you do not move, because this is time. It is going at a slow pace, so you can stop and not.
Is it clear why it's isotropic and isotropic and okay? It's an active stress. We already, yesterday we've seen many types of stresses, right? So this is just, a stress, right? So there is going to be an isotropic part. An isotropic part, that means there is no preferential, all directions will experience the same value of this stress, and then this instead modulates this amount of stress in the particular direction based on, and also accounts for the fact that you have cables and how much are they aligned with each other, basically. Okay, so now this system is very this system basically is very viscous because it's basically made of cells that are basically sticking with each other. Reynolds number is very low, so you can just kill the inertia term, and it's just going to be a static force balance. So the divergence of the active stress, which is what I just defined, it should be balanced by the divergence of the viscous stress. This is what you have also in Navier-Stokes, Laplacian of velocity. Here you have this additional term because the system needs to be compressible on the plane, because cells are basically going into the third dimension. So this is not an incompressible case, and also because the extraembryonic cells are stretching. Okay, so if you account for compressibility, there's going to be this other term that pops up in the passive stress. Okay, and this is just the divergence of the active stress that I just defined. Is it clear? Do you know how to compute the divergence of a stress, guys? Yeah. Okay. Okay, so now what do we need to close this system? To close this system, we need, this is basically two equations, because it's a force balance and B is two-dimensional. And therefore, we need an equation for M and an equation for Q, the nematic order, the nematic tensor. Okay, so now we have to build these two equations. So one is the equation for the myosin dynamic. So myosin is basically now the fraction of activated myosin. So think about it that, each cell, so let's say this is a cell. So this cell inside has myosin, but is not activated. There are all these, protein, actin, myosin, but myosin is not activated. But then it can be recruited to where the cables are, or okay, they could be basically on the junction or internal, etc. And if myosin gets recruited, then you start having active myosin, which generates stresses. Okay, so this M is just the fraction of activated myosin. Goes from 0 to 1. Okay, why I did that? Because this is easy to have, we did that because it's easy to have a dynamic of M that is bounded from 0 to 1, and then I offload the transformation between M to stress to a constant, alpha. Okay. So now if you have a cell that basically has a lot of active myosin and this cell moves, it's going to carry the myosin with it. So basically that means that it's going to be an advection term that carries myosin. Okay. Then there is going to be basically an activation. So you say, okay, I can activate myosin from inactive to active, and I saturate at 1, because at M equal to 1, I cannot activate more. That's all I have available. Okay, so there's going to be an activation term, and then there is going to be a detachment term. This is the detachment between actin and myosin. And people did experiments showing that basically if you have actin and myosin, they are in contact. Depending on how much you are pulling this actin and myosin, depending basically on the tension that is on this actin and myosin, the dissociation rate is tension dependent. The more tension you have, the less you dissociate. This is a so-called catch bond mechanism. It's basically a mechanosensitivity process. Okay, so this basically tells you that the coefficient that governs the detachment, so the detachment rate, is going to be dependent on myosin and has this exponential form, consistent with these experiments. Okay, this M should be tension, but basically if you do a little bit of calculation, you can see that a long time scale, which is basically, you can find it in this paper, a long time scale, basically myosin is a good proxy of tension. So that's basically, it simplifies the equation. Okay, so that's it. So basically this is the detachment, because remember, these cells are moving. They can recruit myosin myosin to become active from their cytoplasm, from the interior of the cell. Then you start stretching this cell because they're moving all together. They feel the effect of other cells, etc. And then when they start stretching, these actin and myosin here that are on these basically junctions, they say, okay, should I dissociate or not? And they dissociate depending on the tension that they feel. Okay, that's basically this term. And the last, and the last bit is that, so my actin and myosin, what they do basically, if you start contracting cables, what happens is that the cables in the cells near you will be under tension. If I contract, I will put my friends under tension, okay? And that basically generates, again, this is basically another derivation that you can see on the paper, but this generates basically an accumulation of myosin in the other cells. Pull, the cells will be more under tension, and with these catch bond mechanisms, basically this generates further increase of myosin in the other cells, that you can actually show that this is equivalent to a diffusion of myosin, effective diffusion of myosin. It's a diffusion modulated by the Q tensor. So this thing can diffuse only if you have cables. That means that the nematic order parameter is different than 0, and then it can diffuse along that direction. It's not a regular diffusion. This myosin cannot diffuse isotropically. It can only diffuse on the cable. Okay, so this is something that you can, it's a directed diffusion along the nematic, cables, or along the direction of the cables.
Like if Q is there, then only.
Yeah, if Q is Q, it's basically the second derivative along the direction of the cable, along phi. So it's basically a directed Laplacian. It's not an isotropic Laplacian.
So it will be projection kind of thing.
Yeah,
yes.
yeah,
Take Laplacian
yeah,
projected.
yeah,
Okay.
yeah. Okay, so this is basically the minimal model for this dynamic, and what we found basically, we found this thing. This is basically now a few years ago. We found basically that if you study the equilibria of this PD, again, what do? You have a PD in this case, not an ODE, look for equilibria. That's all you do, always, right? Because otherwise, what do? You just look at this equation. This is complicated. So if you look at the equilibria of these equations, are basically three fixed points. There is one fixed point that is at very low myosin, activated myosin, basically very low, that is stable. There is one that is also stable at very high myosin, and there is one that is unstable. Just look at this equation that we built in collaboration with the experimental collaborator that told us a lot about this, biology. And, interestingly, we thought, okay, this is exactly what's happening in the extraembryonic. There is no myosin, so it's kind of, it's basically almost with no myosin. This is a passive region that stays passive, unless you are going to overcome this sort of threshold myosin. And if you overcome this threshold myosin, you're going to go to the unstable equilibrium, and this is actually, sure enough, it's what's happening in the attractor. Okay? So basically, we thought that basically an instability of the active stress can actually be a driver of this morphogenetic process. So the embryos may be leveraging this instability of myosin to drive the flow. And in fact, we showed that in the embryo, this prediction was done with the model. We showed actually in the embryo that the myosin increases over time. So it's consistent with this instability. Is this a little bit clear? So depending on where you are, you can either go and stay passive, very little activity, right? But if you overcome a threshold, you're going to go basically towards the higher value of myosin, which is actually what you observe in the attractor or the primitive streak. Okay. We need one more equation for Q. This is basically an equation that is very typical in active nematics, except this term. So Q is basically, again, Q is going to carry the information, two information for every point on your tissue, Eulerian coordinate. You are basically, at any point, there is going to be an average alignment, phi, and a sort of how much all the others are aligned with the mean. Okay, if they're all like this, S is equal to 1. If instead in another region all the things are just randomly aligned, phi is not defined. It's no problem because S is equal to 0, so here Q is equal to 0, which means that sigma active, the anisotropic part, is equal to 0, because that's basically, remember, sigma active was alpha m times identities plus Q. So that means, okay, whenever I don't have aligned cables, I don't have an anisotropic stress. At best, I can just contract isotropically. Okay, so the equation for M, we did it. Is hidden here, so what I just showed. And now you can have, the equation for the cables. I mean, similar to what we've seen before, is a patch of tissue moves, it's going to carry with it Q. So that there is the advection. Then there is going to be two terms that are basically the shear and rigid body rotation. So this is basically standard in active nematics. So, if you basically have that, let's say that, for instance, here the cables are all aligned in one particular direction. If there is a vorticity in this system, of course, all these cables will rotate. Okay, so vorticity, rigid body rotation, vorticity reorients, which is these terms. Okay, and then dd is the rate of strain tensor of the velocity. This S is the nematic order parameter, and this models basically this fact. So basically, if you have basically a shear flow, right, like that, this shear flow is either going to align cables in the direction of largest eigenvector of the shear, or destroy the order. If you start, for instance, from cables initially ordered horizontally, this is going to just flip the order of the cable. Okay, this is also very similar to many of the things that Anupam was telling us yesterday. Actually, you can show that if you remove this term, this guy, remember what are the information that are contained in Q, S, and phi. You can actually, so I think Luca Giomi decomposed using Pauli matrices. Decompose the equation for Q into an equation for S and to an equation for phi, And if you do that, the equation for phi is exactly the Jeffreys equation. So basically, this is exactly the Jeffreys equation for the angle modulated by the nematic order parameter, which is beautiful. It's important because this allows you to play also when the cables or the orientation is not defined, like the defects and stuff like that. Okay, so now the problem is that if you just use these three terms, this system wouldn't reproduce observation, because remember what happens at the streak is that the streak, you remember we observed experimentally that the cables at the later stage at the streak were perpendicular to the streak. But the flow is such that at the streak things are converging. So this is cables, which means basically Q. And the flow here is such that things are coming in and also going out. So this is basically precisely this saddle type scenario. But the problem is that this is an unstable configuration. Okay, so I'm going to flip the cable and I would just turn the cable parallel to this streak. Okay, so this self-induced flow, because this cable contracts, will generate the flow. This is the velocity field. Okay, and this thing would actually self-destroy order at the streak. Okay, so you need a mechanism to sustain order. Okay, and this is what we basically constructed. We said, okay, if I have, there is going to be two terms. So basically, the active mechanism is this one here. It's going to say, if I'm in a region of the system in which I have a little bit of order, that means that S is greater than 0. S greater than 0 means that Q is non-zero. So if I have a little bit of order, and now I start contracting in this direction, so I have a lot of myosin there, so I start contracting in this direction, which is S. What happens, basically, I'm just going to create even more order because I'm going to redistribute myosin and redistribute myosin more in the direction of contraction. So this is basically going to increase the nematic order. Okay, so think about it. Basically, you put, maybe a little bit imprecise, but maybe intuitive way of thinking about it is that if you have a bunch of people with loose ends, if someone pulls, you straighten up all the ends. Okay, that's basically, you can think of this. It's not exactly like that, but that's basically the idea. It's called basically active alignment. So the active alignment is that you start contracting this junction, and then you redistribute myosin in the direction of contraction. So this is where the myosin is going to be more polarized, and therefore, because you're polarizing more in this direction, you're going to increase the nematic order parameter. Okay, so basically we call this active alignment. And then you just have a passive relaxation. So this guy would just, if in absence of all the other guys, this guy would just destroy order, because cells are basically dividing, ingressing, intercalating in a way that you just destroy order. Just is a system basically out of equilibrium. Okay.
What is the small s?
Nematic order parameter.
Small s is the negative.
Yeah.
Prefactor.
Yeah. This is just, if you think of this as an equation for S, this is just decreasing S. Yeah. It's a decay, and this is because if you really think of this system and you look, there are a lot of division, ingression, etc., and these are just things that are randomly happening, and therefore they destroy order. If you would have cables all in a particular direction and cells divide in these regions, you're going to destroy this thing, because you have to reform junctions and stuff like that. Okay.
So S is both space variant and time.
Yeah. Q space and time variant, V space and time variant, M space and time variant.
So usually for decay you use a linear term unless you need to maintain.
There is. Yeah, I mean, we show in the supplementary that basically you can. This is not changing anything. You can pick any functional form that is reasonable. We picked this because it was standard in active nematics to have a quadratic form, and I think the reason was for maintaining S bounded in the active nematic field, not in the bio thing. I think there are a couple of papers from Amin or Luca. So we kept the standard term, but it's insensitive to that form.
Keep it bounded, otherwise linear term will not be fixed.
Yeah.
There is a linear test term that is S and DD. Maybe there is because
there is a linear term.
that's
Yeah.
growth and then that's it, you need a higher order term.
Yeah, but also if you pick it linear, in this case, it doesn't really matter much. Yeah. Okay. Okay. So now we have these three equations. The only thing that we are missing is the initial condition and boundary conditions. Okay. So boundary condition is basically simple. For the velocity, we are just gonna keep from experiments this outward velocity at the edge. You can just pick the velocity from the experiments. Okay, the cells are just pulling at this outer edge, and they pick that as a boundary condition for the velocity. I pick no flux for m and phi and q at this outer boundary, the least invasive boundary condition, because there is not really a known source or fixed value, etc. Okay, so then initial condition, the boundary conditions we are done. We need the initial conditions for m and t, for m and q. Okay, so now we get it from experiments. We know that at the beginning, you remember the disk that I showed you at the beginning? At the beginning, if you really measure myosin, there is more myosin in the posterior where the mesoderm is. That's basically what we found at my initial plot of this disk. So there is going to be higher myosin in the posterior, and if you do the quantification, you see that there are cables aligned in the posterior. There is going to be higher nematic order parameter in the posterior, slightly higher nematic order parameter in the posterior. That's basically our initial condition for the cable and myosin. And we also know in general that there is more myosin in the embryonic region compared to the extraembryonic region. That's all we know, right? And so this is our model initial condition. So the embryo is circularly at the beginning. The boundary of our PDE is here. Here cells will move outward with a fixed velocity. There is more myosin in the embryonic region than in the extraembryonic. We know that. There is also instability. There is a little bit more myosin in the posterior here where the mesoderm is. It's not really good quality, the projection, but you can really show this experimentally. And there is higherly, a little bit of more aligned cable in this posterior region. That's what we basically have from experiments. So what we want to do now, we want to say, okay, from these initial conditions and this PDE, if I solve it for, I don't know, the next 15 hours, can I reproduce the fields that I observe in the embryo in the next 15 hours, okay? And that's what you get. Basically, you get that if you solve this equation, this is going to generate a velocity, which I'm plotting here. So it's basically this embryo, so this model generates a velocity, it's going to generate the vortices, it's going to generate the embryo shape change from a circle to a pear. It's going to generate the extraembryonic that expands, and it's going to generate negative divergence at the streak, which is where cells ingress. Okay, and the interesting thing is that this boundary is not the boundary of our domain. The boundary of our domain is here, fixed. That's basically the boundary of the extraembryonic region. So this means that this model is reproducing a correct or a plausibly correct, basically, distribution of active forces that actually change the shape of the embryo from a circle into a pear. Okay, so this is an interesting observation. First, you can match all these data. You can look at experimental velocities. You get these vortices. You can get negative divergence at the streak, and all the other possible combinations are all in the reference. You can also actually check if this model is reproducing the right distribution of active stress. Okay, so this is saying, okay, now this is the evolution of myosin. Remember, you have myosin is the amount of active stress modulo the constant. So basically, myosin dictates how much active stress you have. So you start from higher myosin in the embryo, no myosin in the extraembryonic, more myosin in the posterior, and later on you see basically that myosin increases because there is an instability, and higher myosin is present, actually develops more at the streak due to the instability, which is exactly what we saw in the experiments. When I showed the myosin distribution, there was this very high cyan concentration at the streak. Now, this is what happens at the final time. The model knew all information at the initial time. Now, if you do the experiment at the final time and you try to measure active myosin, you get this, which is very similar, actually, to that. That's basically the streak. There is a lot of myosin here because these cells are crawling. We got rid of this complexity and we just said, okay, let's actually use just the resulting outward velocity. So otherwise you have to put myosin at the edge, understand the binding to the substrate, etc. So that's basically prediction of this active stress. And also you can actually measure the same for the active stress anisotropy. So now if I want to understand this, so basically so far I showed you this. That's the first plot, and now I want to show you this M alpha Q. Okay, so this is the anisotropic active stress. The magnitude is going to be dictated by m's times the nematic order parameter, which is hidden here, times alpha, but alpha is a constant, so it doesn't really matter. And then there is going to be an information about the orientation, right, which is also hidden in Q. Okay, so that's what you see here. So a little bit of orientation in the posterior, more myosin in the posterior, and then you let it go, and basically you see basically how the cables will reorient. There is going to be a lot of, anisotropic active stress at the streak, and the cables are mostly aligned perpendicular to the streak. Okay, so this is basically M times S. This is the orientation of the cable. If you do the experiments, you see something very similar. So basically there is a lot of alignment of the cables perpendicular to the streak. Okay, so we're happy to compare basically velocities or models from, from experiments and model, also, the active stress. But what we wanted to do, we wanted to reproduce the attractors and the repellers, okay? So now we said, okay, what I can do, I can just use this velocity field and advect a Lagrangian grid. So I start with a uniform grid, and then the later stage I get basically this distribution of the grid. Okay, so this is what I get from the velocity of the model, and you can do the same from the velocity of the experiment, and you get something similar. Okay, so that's basically the embryo has changed shape. Okay, and now you can compute the repellers. If I compute the repellers from the model, there are going to be two repellers. One that is very strong, is separating the anterior part and the posterior part of the streak. That's repeller number 2, and this is repeller number 1. That is separating extraembryonic with embryonic, and of course is more demarcated where the pair actually shrinks. And you can see it from here that there is a larger stretching of these rectangles compared to here. Okay, so there is still going to be a relative separation. You see basically that this guy is expanding, this guy is contracting, so there is going to be basically a separation, but this separation is higher in the posterior because the pair is basically shrinking. Compute the same for the experimental velocity and you get something similar. So you get repeller number 1 and repeller number 2. Okay? Okay, so now we want to understand how do they form. Can I actually deconstruct these repellers and understand what forces are, or what combination of forces are generating those, okay? And can we control them? Okay, so this is now going to be the uncontrolled or the wild type, and now I'm going to start deconstructing these repellers. So for repeller number 2, so we wanted to eliminate repeller number 2. And we thought that basically repeller number 2 intuitively is associated with the myosin, with the mesoderm that starts in the posterior, with the guys that do active intercalation. They will generate basically convergent extension and they will elongate the embryo. So what we did basically in the model, we just left everything unaltered, but we basically eliminated this myosin in the posterior, this myosin bump in the posterior. So remember that at the beginning there was an additional myosin bump in the posterior. So what we did, just let me eliminate this initial condition, which is eliminating the mesoderm, and solve the problem as is. And if you solve it, basically the model predicts that the embryo stays circular, and now the repeller number 1 becomes circularly symmetric and actually separates the extraembryonic cells from the embryonic cells. Because I removed basically this myosin, I let the model run, and the model predicts that there is going to be a circularly symmetric repeller and the embryo keeps a circular shape. Okay, same parameters, the only thing I just changed is this initial condition. Now the collaborators basically they have an FGF receptor inhibitor. So if you basically add this FGF receptor inhibitor, you block mesoderm induction, so therefore you eliminate mesoderm cells. Okay, so you go to the embryo, do this drug, and you do the same experiments, and the embryo with this drug, no mesoderm, stays circular and develops a repeller that is circularly symmetric compared to the wild type. Okay, so basically this means basically the repeller number 2 is implicated with embryo shape change. Okay, because now the embryo is not changing shape. Okay, so that's basically what we were able to show. Now we say, okay, can we actually eliminate repeller number 1 and keep repeller number 2? For repeller number 1, remember, is this that separates embryonic with extra-embryonic. We said, okay, this is actually caused by a competition between these cells that are pulling and stretching the embryo, the extra-embryonic cells that are basically pulling away up, pulling out, and the myosin contractility inside the embryo. Because, you know, isotropic myosin contracts, so you have a force that contracts, another one that pulls, their balance will generate this repeller. Okay, so what we did basically in the model, we said, okay, I'm gonna just turn off the boundary conditions of the velocity, and I leave everything unaltered. Okay, I do that, and then the model reproduces embryo shape change, but in this case, the size of the embryo decreased because basically there is no pulling from these outermost cells. Okay, so this is the embryo basically retains shape change, develops repeller number 2, but eliminates repeller number 1. Okay, and our collaborator actually, this experiment was hard to do. What he was able to do to mimic this was basically burning or cauterizing the extra-embryonic regions, or where actually the vitelline membrane, which is the regions where these cells attach, and therefore if you burn them, they cannot attach anymore, so this really imposes V equal to 0 experimentally. Yeah.
What exactly?
They burned the vitelline membrane. So remember I showed you that the outermost region of this system attaches to this substrate, and that's where cells start moving. Okay, so basically it burned this part, so these cells basically cannot go anywhere. It's just a wall. Okay, that's basically the black part here.
So if I understand, so the velocity has been set 0 when you said CB.
No, V equal to 0, just a boundary condition. We just replace that. The rest is the same of the model.
Okay, that's right, because if you treat with some drug, then it becomes tricky. Then it can affect the velocity.
Yeah, there is no way you can, it's also, people have tried to, this is a very challenging system because people have tried to basically cut part of the cells that are actually crawling. They are so smart that they redifferentiate into cells that crawl, and then they keep pulling again. And this happens basically very asymmetrically around the embryo. Some cells will take less time to redifferentiate, so you're going to have Guillermo did basically a very smart experiment, I think, that he said he burned it, and this was really imposing the velocity as in the model. Okay, so now this basically retains repeller number 2 and eliminated repeller number 1. Okay, but the embryo became smaller. Okay, so we basically thought the repeller number 1 is important to regulate, is a contributor to regulate embryo shape, size. Okay, and of course you can do basically the same. You can combine the two treatments, and you can have basically nothing. You have 0 repellers both in the model and in the experiment. So if you basically block epibole and also eliminate mesoderm, nothing happens basically in this system. So this basically was interesting because we sort of just guided by these coherent structures, we were able to understand that repeller 1 is arising from a tug of war between the embryo constriction due to myosin and the extraembryonic crawling. Okay, and this is important to regulate embryo size, so the size of the embryo. Why embryo size is important? There are several speculations. We have basically a comment in the supplementary section, but embryo size is very important if you have a patterning mechanism. If you have patterning equations or processes to say, okay, now I have to have adduction reaction diffusion, etc. If you mess up the size, you're gonna have very different Turing patterning or patterning situation.
Number of somites and all.
Yeah, everything, right? This is, so that's our speculation. Of course, it's very hard to test. It's a sort of an evolutionary question. Say why the embryo would like to regulate its size.
These guys have done that. They cut the embryo, so effectively you reduce the size. You can cut the embryo and then you can look at the growth. Means it is not exactly same.
Yeah.
They see that the structures differ.
No, for sure. It's very likely that size is going to affect patterning. And repeller number 2 is instead arising from the active intercalation of the mesoderm and is associated with the embryo shape. That's the guy that transforms a disc into a pair. And this was basically, it looks maybe a little bit straightforward a posteriori, but it was really driven by these attractors and repellers because we were after them. So, okay, you know, it's cool. I mean, I understand how they form. Can we actually control them? And then we discovered this modularity. Because, for instance, it wasn't clear, at least from my understanding on the literature, that when you block mesoderm, people think in this community that everything stops. But instead, the active contractility of myosin, the isotropic myosin in the embryo, stayed to regulate the embryo size. That was a very surprising biological new result. And you know, the fact also that you can knock mesoderm and the embryo still retains its size is a cool thing. So basically, we're able to control, understand how these repellers form, and also control them independently. So yeah.
So for the second case where you remove the repeller one, so basically you're setting that at those points the embryo is not going outside the overall.
Yeah.
So, okay, so how does that affect the size? Basically, just to get.
Because think about it. Basically, think about you know, a disk of cells. These cells are contracting, but you're going to experience a pulling force. The pulling force would like to stretch the embryo, so it would like to actually increase the area.
Okay. So you're trying to contract, but someone is pulling, so you overall grow.
Yeah, but
stretching is not there, so you will just shrink and.
Yeah, exactly. Yeah. That's the thing. Of course, it's more complicated than that because basically, yeah. Okay, so this is basically control of repellers. And a few years ago, actually, this was a work that we did also with Maha when I was still, you know, in Boston. We were also able to control the attractor in a very exotic way. So basically, this is the attractor, right, the streak. That's the model. And then we were able to control the attractor in many different ways. You know, this is basically spontaneous twin development. So the attractor now is made by two streaks that try to fuse. You can get it in the experiment and in the model. We were able actually to control this attractor in the cheek from a line to a ring. Okay, so basically, let me show you. So this is basically the model and the experiment. Again, what we did here is the same stuff that we did basically here. We played with the initial conditions and some parameters that are biologically interpretable, and you know, you can implement them in the lab. And with the same mechanical model or your mechanochemical model, you can actually reproduce very exotic perturbations. So to give you a more visual interpretation, the cheek develops this deformation pattern, but we were able to manipulate the cheek in a way that develops this deformation pattern, in which basically now instead of having an anterior-posterior axis, a streak, you're going to have a ring, and the ingression is on the ring, okay? Or even into a point attractor, okay? So, you know, you can also transform this and have that everything contracts to the center, you know, both in the model and the experiment. Okay, so this is basically controlling attractors and repellers. I'll do a mini summary and then I'll add 2 more slides. So what did we actually learn is that these coherent structures allow you to compress spatiotemporal velocity into discrete units, attractors and repellers. Then we were able to build an active matter model that can explain these flows, the active stress patterns, and you know, this dynamic embryo geometry, size and shape in a modular way. And then we learned that, you know, repellers, they control basically embryo size. Repeller 1, the repeller 2 controls embryo shape. And this is basically an interesting way to discover modularity in these systems. So this is basically Keys, you know, is the guy who basically taught me the little biology I know, but all the credit goes to Keys, and also Guillermo was a student with Keys at the beginning and now is a postdoc in Cambridge, and Alex is a student in the group. So basically this to say that attractors and repellers, they are not only a way to compress and classify flows, but they can also guide you to build models, okay, and design perturbations, etc. I want to also, you know, this is basically advertise a couple of more things. So if you want to compute attractors and repellers now on curved surfaces, dynamic surfaces, so this is basically beating zebrafish heart, experimental images. This is a pancreatic spheroid. This is basically active nematics on exotic, you know, curved surfaces. You can now compute all these structures when your flows on, you know, these different geometries, and we also have, you know, the codes available. They are basically, I think, we share it with you. I think that's it. That's all I have for the morning. Yeah.
And the last is on the curved surface.
Yeah. Yeah.
So this is on what surface? It's what system that represents?
This is to show basically that, this is to show that they can compute. So this is the simulations were from the group of David Santillan at UC San Diego, and they now can solve active kinematic flows on curves on any geometry. Yeah. So now you can, I mean, the interesting thing, I don't know where did it go, but the interesting thing that now you can start looking at, yeah. It's fine, I don't need the slides, but in general, you can now start understanding, so there are a couple of interesting things. First of all, in organogenesis, you have flows on curved surfaces that are themselves evolving, so how do you do this analysis? And the other thing is that now, when you have curvature and curved surfaces, you can also start understanding what's the effect of the curvature of the surface on, I don't know, the flows, the strain, and stuff like that. And you can also use this for doing control. So because on particular curved surfaces, the motion of topological defects are of a particular type. This will steer the motion of material transport, etc. So this is now a way to attack basically these problems. Yeah. And one more thing, guys, in the talk that is gonna be during the conference, I will talk about how these attractors and repellers are also important for understanding advection reaction diffusion. So if you have basically advection reaction diffusion on top of flows, which is very typical, for instance, in development, but in many systems that are deforming, you will see basically that very elegantly these attractors and repellers and Cauchy green will modulate diffusive fluxes. Okay, so this is something I think I'll discuss on Monday or Tuesday. Yeah. Okay.
Thank you.
Okay.
Questions?
That's good, yeah. If there are no questions, it was clear, or you're shy.
I think because for many of them, maybe they're seeing this kind of system for the first time. So
Yeah. But I mean, basically, it's a beautiful connection with the equations that Novam was saying, basically, of the similar family. Yeah.
But important things one should notice that, the experiments are how beautifully done, but how to interpret them, right?
Yeah.
How to dissect each part. I mean, that's how beautifully it was done. Okay, what is responsible for this part, like one kind of repeller, what is responsible for other kind of repeller. Also designing it. I mean, this takes a lot of thinking and a lot of time.
Yeah.
Of course, they managed to figure it out.
It took, I mean, this basically, we've been working on this system for a few years now. Yeah. All right, that's
okay,
good.
Let's take a break. We'll go for tea, coffee, and then we'll come back at 11:30.
Yeah.
So backward in time also the system will retain its original state.
Sorry?
Backward, if I apply backward in time, will the system reach to its original state?
Well, which system? Tell me.
The attractors and repellers that we have seen.
Yeah, so we were discussing this yesterday. So if you, yes, because basically these are just accounting for motion. Okay. Now the way to think about it, maybe after the discussion of yesterday, is that if you give a velocity field, and you pick the initial condition, someone gives you a velocity field, let's say the measured velocity field of the embryo, okay, of that embryo, okay, and you advect an initial position later in time, and then you advect it back in the same time interval, you go to the same initial position. Okay, so that's basically because you have a velocity field that is deterministic. Now, the reversibility that creates confusion is that eventually you are thinking about something that advects and diffuse, like a parcel that advects and also diffuse. Okay?
The alignment also written exactly back to its original state.
Yeah, the alignment, I don't know. So you're talking about, is this equation, which equation? So the PDEs are not reversible. but the PDEs are describing the evolution of the velocity or the Q tensor.
Velocities are, we are getting that from PIV, right? The data that we have.
Yeah, so you are talking about the model or the experiments?
The model.
Okay, the model. The model also predicts the velocity because there was the model and the experiment, right?
But we have three equations, right? One is with the governing M, and
Yeah.
we have one with the governing Q,
Yeah,
and one is the force balance.
yeah. That gives you B.
That gives us V.
Yeah.
But S is also unknown, right?
S is the nematic. Yeah, SS is in Q.
But we have only three conditions, right?
Yeah. No. Q has 2 degrees of freedom basically. S and the angle. So we have 4 equations for unknowns. Yeah, we need to have 4 equations for unknowns. Yeah. I mean, 2 velocity, 1 m, and q that has s and the angle. So we are not input.
Q and S are kind of interdependent.
Yeah, they are interdependent. Yeah.
You get it from the director field. Like you define L, and if you, so you can get a. So of course, the vector sum could be 0. That means there is no ordering, but still magnitude is there. So that magnitude gives you the isotropic part, and the vector sum, if it is finite, then that is going to give you the ordered structure, the anisotropic part, and then you break them in 2 parts. So. And you have, yeah, that's how it comes. And then you have to maintain, like, Q should be, an isotropic part should be, the trace of anisotropic part should be 0. And how should be
Yeah, that's all built in. Yeah.
So once you take care of those things, you will start getting the magnitudes for the isotropic and anisotropic part. So if we know the structure how it should be, we have the director field, like the coarse grain structure, that the yellow color vectors were there, right? So that's your director field.
Yeah, from the experiments. Yeah.
And of course you can do the same for
the model. Yeah.
So yeah, that's how you. But yeah.
Okay.
I'm not well with pneumatic fields.
Yeah. No, it's a cute system. Yeah. All right.
Okay, very good. Good. You are following?
Are you following or are you hanging out there in the south? Okay.
Because, but again, I said that if you do not understand something, you can
Okay, let's see if this works. Thank you. I'm gonna wait for Anupam and then, yeah.
Then started out and finished the mission. And then they should be
That is just the secret. Yeah, but I mean, you can take anything, but it is still good.
Okay, guys, let's restart. Hey, guys in the south there.
Yeah.
How's it going? Okay. So we've seen an example before on, let's say, the forces that can generate some type of coherent structures, and how you can use it to develop models and control these systems. This is a different type of coherent structures, and the application is related to search and rescue. And I'll try to connect also with what we wrote on the board. So hopefully it's going to be more, easy, to link. And, please stop me. I don't have, slides for 1.5 hours because the idea is to have it more interactive. Okay, so what happens during search and rescue operation is the following. So there is basically, there is a call to the Coast Guard or whatever, unit is or agency is responsible for rescuing whatever is floating or sinking. And then the Coast Guard or this agency has a set of limited assets and, let's say, helicopters, boats usually. And you have to react very quickly. At least for search and rescue operation, the relevant time scales are less than 6 hours. And usually, future and past data, they are available, but what happens in the context of search and rescue is that whenever there is a call, basically, let's say there is a call here. Let's say this is the US. Let's say there is basically a call here. Let's say the plane lost communication in this region, and there is a certain amount of time by the time boats and helicopters can get near this area, and then you can sort of identify this sort of uncertain initial area. And then what happens is that there are going to be some ocean models available in the globe, depending on where you are, that are, for instance, I don't know, good in this area. There are others that are good in this area, others that are good in the open ocean, etc. So then they pick some model, okay, or they do some averaging of this model, and then sometimes the idea is that, of course, these models are not super accurate because you cannot really resolve the actual ocean velocity. This is very complicated. So then what happens is that ideally they go with some helicopters here, and they basically they deploy some drifters in the water. These drifters move, and as they move, this data is assimilated into the model so that you're going to have a surrogate model that is more accurate in this region and at this time. Okay? So in general, what happens is that you have model in the future, in the past, but the most accurate data is the data that is happening now in that region because it's augmented with local observation. For instance, they say, let me actually assimilate temperature information, drifter trajectory, etc. Okay, so that's the challenge, and this is basically similar challenges when oil spill happens. And especially in the Mediterranean recently, in the last few years, thousands of people died for crossing the Mediterranean. There was a refugee crisis. Okay, so basically these properties or constraints for this particular application suggest that Lagrangian methods are not really ideal because those require integration of trajectories, and we saw that basically future and past data are not ideal. They are not as accurate. Okay, and eventually you want to start providing information to the Coast Guard where to go immediately, as soon as possible, because 6 hours is very small time interval. Okay, so what is basically the standard approach of search and rescue is that let's have a little bit of notation. So fluid particles, they will obey this differential equation, x dot equal to v of x at t. That's the velocity of the fluid. Floating objects, usually you can write them in this form. All the equations that Anupam was discussing, you can cast them in this form where epsilon is a small parameter. Anupam was calling it capital V yesterday. Is the speed of active motility divided by the speed of the flow. Okay, so then you can say, okay, this is the ocean model velocity. These are a lot of things that you don't know typically. This could be, I don't know, the current windage in that particular region in that particular time. I don't know, the inertia of the objects, the shape. There is basically so there are lee waves or currents on the surface of the ocean that will also affect the transport on top of the advection from the water, but those are basically typically unknown. Okay, you cannot really start resolving that and doing Monte Carlo simulation because you really have to react very quickly. So what they do, the current approach is you do that, then they run ensembles of all these possible uncertain parameters. They do Monte Carlo. Monte Carlo provides you a probability map of where things would accumulate most likely, and then on top of this probability map, you're gonna start having basically scanning of for helicopter or boats. You have to scan regions of high probability map. Okay, and then you say you go there, you didn't find it, update the probability map with a Bayesian inference. And what happens basically is that if you are doing a search with Bayesian updates, and in a simple setting, usually you would expect that this probability map becomes more peaked over time. Eventually, it's okay. I'm gonna go very, I'm gonna increase I'm gonna decrease the uncertainty of where the objects could be, in general. However, in the classical example of search and rescue, this is not the case because you have the ocean, which is a very chaotic system that is forcing the search. So you start updating this probability, and it becomes even more diffused. So it's a very challenging thing because it's okay. Where do I go? So there are a few problems. This is very slow. Okay, it doesn't really necessarily converge because the ocean surface is forcing this system. So therefore, it's not like you're looking for a fixed source in a very steady environment. So the more information I accumulate, the more sure I am where the source will be after I accumulate information. This is not the case because it's very time dependent. And the other thing is that once you have this probability map, you are left with a two-dimensional probability map, and then you have to do a further scanning. You have to do some pattern scanning within this probability map to see where to go, to scan this area. So this is basically a little bit of structurally, not favorable to have this procedure. So then we said, okay, can we actually identify or define the notion of short time attractors and see, which are very quick to compute because short times Eulerian coherent structures, so Eulerian you don't even have to integrate trajectories, and these are so-called in the terminology, in the language, they are codimension 1 structures, the ridge of the FTLE or stuff like that. The codimension 1 means basically they are one dimension less than the dimension of the phase space of the velocity. Two-dimensional flows, codimension 1 structure are lines. Okay, one-dimensional lines or curves. In 3D, they are surfaces. So this is beautiful because basically now you don't have to search volumes or areas, you go with lines. So you have a technique that already returns you something that is very prone to be explored because it's easier to explore a line rather than an area. Okay, it's okay. Can we do that? Okay, so the hope here is the following. So the hope is that, okay, we cannot really resolve this uncertainty because, if you really know detailed information of G, I can construct traps or transient attracting profiles. These are also called Eulerian attracting coherent structures. We call it traps for this basic application. This is transient attracting profiles. The idea is that you have velocity available. If you give me information about G, I can also modify the definition of traps and say, okay, if you're looking for inertial particles, I'll tell you where they come. Or if you're looking for particles of this particular shape, I'm just going to consider this as the new velocity field and define the traps on that. But the problem is that you don't really know G. So the question we ask is, can I extract the most robust information of the velocity field that will likely have an impact also on the trajectory of floating objects over a very short time? Okay? Because basically epsilon is small. Okay? So there's a very important distinction here. So Anupam showed you, Anupam equations were very similar to this. In general, also the equations for phytoplankton, for everything is of this form. It's a velocity field of the fluid plus a small parameter times some other functions. So this is always small. Now, if you're interested in the long-term behavior of things, this small parameter can make a difference. But over a very short time, it doesn't. because this is going to be velocity plus a small correction. Okay, so it's very important to understand Lagrangian versus Eulerian versus asymptotic. Okay. Okay. So what we want to do is this. So basically, things fall in particular region. And because the initial condition is uncertain, so basically you're never going to know where you land in this unknown domain, we said, I'm not going to insist on trying to resolve the uncertainty to initial condition because it's a lost game. On the other hand, what we want to do, we want to say, where are the most attracting regions over a very short time in this dynamical system, right? So basically, pictorially, this is what we think. So we think this is an uncertain location. Now I want to understand, so this would be a short-time repeller, and then eventually there is going to be a short-time attractor, okay? And you can actually have maps of these structures in the ocean as they evolve, okay? So there is this model velocity field. These are instantaneously to compute. We are going to see how we are going to compute it. And then, to be honest, I don't really care about predicting all this trajectory. I just want to understand where are the most attracting region. It's like the way I think about it is more, imagine that you have a metal table and there are some hidden magnets below that, someone can move, and they throw a coin. Predicting the trajectory of the coin is going to be very hard because you see this magnet, the dynamics is very transient on the ocean surface, etc. But if I have the stronger magnet, most likely I'm going to get rich with the coin. Right? I'm going to get the coin, right? So this is a little bit of an intuition of forget about reconstructing the full trajectories, focus on where are the most attracting regions in this system over a very short time. Very short time in this case means that you can compute them with a single snapshot of the velocity field. One snapshot, compute where are the Eulerian attractors or traps, and then when there is the new batch of velocity available, you recompute it and you see where it evolved. Okay. Okay, so basically this is the problem. So you can think of this as there is a set of possible initial conditions, which is like this. And now, they will always go to usually in different locations, but eventually I want to identify the attractors or the short-term attractors that will most likely attract whatever is floating, regardless of the specific trajectory of each one of them, because I don't know which trajectory is going to be the relevant one. Okay. And this is what I already told you. Can we actually build traps based on fluid velocities such that they will attract floating objects? Okay, so how do we define these traps? Okay, so basically this is a little bit of the slides that I used at the beginning. So, we basically passed from asymptotic attractors in the book. This is basically you anchor yourself to a fixed point, you do this construction with center manifold theory, you can compute unstable manifold, they only work for steady velocity fields, so this would be impossible. And also, if you wait for infinite time, there's no point of doing search and rescue. So the question is, okay, then there was basically the finite time setting, or the Lagrangian coherent structure. Here we are bringing this limit to the opposite. What happens over a very short time? Okay, and an interesting thing that you're going to see is that even in this case, if you just look at the streamlines of the velocity, you don't get the right answer. Okay, we're going to see why. So that's what we want to do. Now we want to identify where are these objects, these curves, red curves, let's say the attractors, the short-time attractors that will actually attract nearby particles or floating objects over a very short time, infinitesimally short time. Okay, so this is basically the other day at the course. We have basically a velocity field, a flow map, just the final position of a particle. Then you have the Cauchy-Green strain tensor. And remember what really does the Cauchy-Green strain tensor? Just encodes all the Lagrangian deformation of any particular material fiber, zeta 0. Okay. Now let's use what we learned the other day. So now we are saying, so remember, Cauchy-Green is something like, so Cauchy-Green is df transpose times df. Okay, so this encodes all the possible Lagrangian deformation that you start at any point you start, initial fluid configuration. This is, you're imagining a ball of initial conditions here, and for any one of the points on this ball, Cauchy-Green tells you how it's going to deform. Okay, so now this of course is Lagrangian and requires a time interval t0 t. What happens if I want to understand, so t0 t, or eventually you can write this as t0 to t0 plus capital T. What happens if I now want to understand in the limit of t going to 0? What happens in this case? Okay. Well, what do you usually do when you want to understand? You have a function of time, and now you want to understand a limit for t going to 0. Taylor expansion. Okay, so let's do it. So basically, you're gonna get a c at t0 plus, let's say, t of x0. Well, yeah, basically, let's do this. I don't want to repeat this, but okay, what's going to happen? So the first one is evaluating the point at t equal to 0. Okay, what is it? The Cauchy-Green at t equal to 0? T equal to t0. This is basically, now this is going to be the leading order term, which is when t is equal to t0, which means basically t equal to 0. How is that transforming? Okay, df t0 t0, what is it? What was the initial condition of the equation of variation? Identity. So f, remember f is equal to x0 plus the integral of the velocity along trajectory. Okay, from t0 to t. You have to hold this tau, it doesn't matter. This is t0 to tau, right? So basically, the position at t equal to t0 is going to be equal to the initial position. Therefore, when you take this, when you eat this with a gradient, you're going to get that this is going to be the gradient of x0 with respect to x0 plus the gradient of this guy. This you can basically remove because basically this is a t equal to t0, t equal to t0. So basically this is telling you that the derivative of x0 of f t0 t0 is equal to identity. Okay. Okay, so what is this? This is basically identity transpose times identity is identity. Okay, plus, now we need the derivative of Cauchy-Green with respect to time. So we basically need the time derivative of Cauchy-Green with respect to time evaluated at t equal to t0 times delta t plus order delta t squared. Right? So, fine. What is this? What is this now? It's c dot. Okay. So let's do c dot. This is the total derivative. Where this means total derivative. Okay. So then we have to do c dot is equal to what? We just expand the product. So it's going to be now df trans dot times df plus df transpose times df dot. They are all total derivatives, right? Okay, what is now df dot? The most beautiful equation we wrote the other day, that's the equation of variation. Remember? So df dot is equal to the gradient of the velocity field, we call it v here. So this is going to be the gradient, the Eulerian gradient of the velocity field v along trajectory, so t0, t, x0, t, times the deformation gradient. Remember, this is what we did the other day. Okay. So now let's plug it in here. So now I use these two together, and I get here that c dot is equal, is going to be equal to df transpose. So this is going to be df transpose times dv transpose. I just transpose this guy times df. So this is basically df transpose. I'm going to get the transpose of this times df plus this guy is going to get df transpose times df dot, which is again the same, dxv times df. Right? Okay, but now we have to evaluate this guy at the initial, at t equal to t0. Okay, when you evaluate that at t equal to t0, what do we get? Okay, let's evaluate all of them at t equal to t0. df at t equal to t0 equal to identity. Cool, so this is gone, so this goes to 1, this goes to 1, this goes to 1, this goes to 1. So c dot at t equal to t0 is equal to dx v. Of course, when you evaluate f at t equal to t0 is at x0. So this holds at any point. I mean, you can call them the way you want. You can call them x0 or x comma t plus dxv x comma t. Okay, and what is that? It's twice the rate of strain. Yeah. So basically, we learned that, which is not surprising though, right? So what is governing stretching over a finite time is Cauchy green, and the infinitesimally short time is the rate of strain. So basically, we learned that basically c t0 t of x0 is equal t0 plus t in this limit of t going to t0 for t going to 0 is equal to identity plus twice the rate of strain tensor. I want to call the rate of strain tensor S xt. I mean, I can just call this x0 t0 delta t plus order delta t squared. Okay, so basically, of course, there is going to be, at the initial condition, nothing is deformed, so we're going to start with your initial fiber x0 plus a little order correction, first order correction that is going to be set by the rate of strain tensor. Okay, so now if we want to understand attraction or repulsion, actually attraction and repulsion rates, you have to play with S and not C. That's the leading order correction is all included in the rate of strain tensor. Is this relatively clear? You can redo the derivation independently, but it's basically that's what you get. So when you differentiate C with respect to time and evaluate it at t equal to t0, and you get that this is basically the leading order correction, and that's the rate of strain tensor. And therefore now basically there are two objective tensor fields. For any initial condition, initial configuration, you can say, okay, if I start at x0 and I want to understand what happens over, I don't know, a week, you compute the Cauchy green that you already did in your computer. Or if you say, okay, I really want to understand what happens in a very short time from this time, then it's all the information are in the rate of strain tensor. And these two tensors are both frame independent. They are objective. They check all the right requirements basically to identify material deformations of a continuum. In this case, is basically fluids. Okay, fine. Okay. Okay. So now we want to understand. Okay, now we have the same problem we had before. We have a tensor. It's a little harder to visualize this tensor. What do we do? Okay, so let's try to. So this is what we did at with George. This was basically my first paper of the PhD. We said, let's try to identify what are the most attracting curves in a dynamical system over a very short time. So to do that, you basically parameterize. So you're in 2D, in this case. Now you parameterize a curve. Think about this is the ocean, and now parameterize a curve x of s. This is a vector. X of s is a vector. So, you know, you're going to have x1 of s, x2 of s, which is equal to x of s. I don't want to write this, but it's implicit that this is a vector. S is just some parameterization, could be arc length or whatever you want. It doesn't have to be an arc length. So the more you scan S from 0 to sigma, which is the length of the curve, and this is going to trace a curve in 2D, right? So this is going to be x of s1, x of s2, x of s3, so this is your curve, gamma. Okay, and this curve is going to have a normal, so a tangent vector, which is dx ds, which I'm going to call it x prime, and then I can also define a normal, which is x prime rotated by 90 degrees, n of s. So now I have this curve. I don't know where these curves are. Eventually, I want to go chase these curves that are the most attracting in my dynamical system phase space, but I have to set up the problem. So I'm going to look for curves, convention one structures, that you have to parameterize them. So you have to pick parameterization x of s, and you're going to need tangent direction and a normal direction. So this is basically the parameterization. It's just the mathematics is not important. And then what you want to do, you want to say, okay, now I have this curve, and I know that there is going to be the stretching rates of every neighboring points on these curves will be governed by the rate of strain. That's what governs the material deformation. So what I want to understand now, I want to say, okay, imagine that I have a normal distribution of vectors to these curves, and I want to understand how much do I attract to this curve, because that's basically my notion of short-time attraction. So what I want to do now, I want to say, okay, how much the normal vector to this curve shrinks. That's a measure of pointwise attraction. That's basically this quantity. The normal repulsion rate or attraction rate is the same. You say, okay, I'm going to have r times x prime is the normal at any point. Okay. Then I'm going to say I multiply it by s. This is basically the s times this normal, and then I project it on n, which is r times x prime. I'm just I'm gonna put now the r inside in this quadratic form. So I'm gonna have basically a functional that is gonna tell me how much this normal fiber will stretch or shrink. Okay, is it clear? Okay, that's basically exactly. And then I say, okay, this is stretching or shrinking, and I just want to divide by the norm of my parameterization. So if you pick unit parameterization, you can set this to 1. Okay? So this is going to tell you are in your phase space. So something, let's say this is the ocean. Then it's going to be longitude and latitude. And now you're saying, okay, I don't know what these curves are. I'll tell you how to compute these curves. But, for any curve, gamma, I'm going to tell you at any point, tell me the normal to this curve, and I want to interrogate, because there is a flow field in the background, given the velocity field. So here. Okay, this velocity field is going to have basically a rate of strain tensor that I know governs stretching rates or deformation rates over a very short time. And then I want to say, okay, this curve, this point, is it going to experience a stretching rate or a contraction rate? Okay, and this quantity is just q prime, q dot, little q dot, okay? This quantity depends on the properties of the flow, S, depends on where you are, point, in your basically dynamics, it depends on where you are, and also where you are, it depends on which fiber do you want to interrogate, which is the normal to the curve. Because we know that, as we've seen from Jeffrey's equation, we know that if you start changing this direction, you're going to have different shear contribution or different stretching rate contribution. Fine. So basically, that's what happens point-wise. Now I say, okay, let me actually consider an average of this quantity on some curve gamma. That's my functional. It's the average normal repulsion rate or stretching rate, if you will. Basically, it's just 1 over the length of the curve, integration over gamma of this little quantity. Okay, so this is now a functional that depends on, of course it's going to depend on the velocity field, etc., but the velocity field is given to us. So the only thing that is unknown is where is gamma? Where are the curves gamma that, given this velocity, will spot the regions that are the most attracting regions, right? So to do that, so basically now you say, okay, I'm going to define these traps as the minimizers of Q. So those are basically, they are going to be the curves that will maximize the attraction rate, which is the opposite of repulsion rate. Okay. So when you have this problem, it's a calculus of variation problem. You have a functional, and you look for the extremizers of this functional. Okay. Basically, you take the first variation, the variational derivative of this functional with respect to the function, basically gamma, which is you have to parameterize perturbation to this x of s, and you're always going to get a boundary term and an integral term. Okay, and then you can solve the integral term, and this is going to give you basically the Euler-Lagrange equation. Is going to be basically a PDE, an ODE, for the curve x of s that is going to actually maximize, or at least as a first order, a zero first order variation, so it's basically an extremizers of this problem. Okay, so basically this is the philosophy, and this is going back to the lecture number 1. You know, here the coherent structure really depends on what you're looking for, so you have to build your functional that really reflects your needs. Okay, this functional has the right players in it, because I know that I want something that is frame invariant. I wanted something that's not gonna change if I just have arbitrary choice of the reference frame. Okay, so then you pick the right tensors. So if here I would see the spin tensor, the spin tensor is very frame dependent. So I would have said, hey, I don't like, so either I'm doing a mistake, or the function that I'm trying to optimize is not representing material deformation. Okay, so this has the right ingredient, rate of strain tensor, and then you build the mathematical function that describes mathematically what you want. Then you solve this problem, this is just a mathematical problem, and then you get the result. The result is the following. So in any dynamical system, okay, this could be doesn't have to be any fluid. It's just a dynamical system that it basically goes by x dot equal to f of x in t. Okay, so that's x can be in any dimension. The problem is that then it's a little bit more complicated when you go in higher dimensions, because it's not going to be a curve, but it's going to be surfaces or hypersurfaces. But philosophy is the same. So if you are in any dynamical system and you want to ask what are the instantaneously most attracting curve at a particular time, they are the extremizers of the problem we discussed before. They are computable. This is a result of what you can get from the irrational problem. They are computable from the Eulerian rate of strain tensor, symmetric part of the velocity gradient. They are tangent to the dominant eigenvector of the rate of strain tensor. So you compute the rate of strain tensor everywhere, like you did it for Cauchy-Green. Now it's another tensor, it's symmetric, similarly, so you can use the same code. And then you compute the eigenvector, and then what you do, so this eigenvector now is a director field, is not a vector field, but you can still integrate director fields. Okay, you have to do some correction, because the vector field, like the velocity, tells you when you start here, it tells you where to go. With the director field, you can go either in this direction or in this direction plus pi. It's fine. You can do a little bit of correction and then still integrate director fields. This is how people compute nematic texture in liquid crystals, right? So you just integrate the nematic director back and forth. So you basically integrate this director field in the direction, so for the director field specified by the largest eigenvalue of the rate of strain. And where do you start your integration? At the smallest, the minima of the smallest eigenvalue of the rate of strain. So let me give you a picture here, and then we're gonna go with the examples that we have later. So basically, the results of this calculus variation exercise, which is the optimizers that we're looking for, they are as follows, basically. This is the ocean, okay, and let's say for a particular time t, this is, longitude and latitude. Here there is a velocity field. Then you compute from this velocity field the rate of strain tensor, which is dv transpose plus dv divided by 2. And then here you just compute the eigenvalues, like we call it S1, the smaller eigenvalue, S2 is the largest eigenvalue, E1 is the largest eigenvector, the smallest eigenvector field, E2 is the largest eigenvector field. So we are saying, look at S1. So S1 is going to be a surface, right? Like we did it for the FTD. So let's say that S1, let's say is going to look something like this, and then whatever, right? So, okay, so this is S1. This is the value of S1 as a function of x and t, this particular x and t. So you say, yeah, look for minimum of S1. This is a minimum of S1. This marks a very important point in the fluid. We are going to understand why. And then on this domain, which I like you can do it with Cauchy-Green, there is going to be another vector direction field, which is the E2 field. This would be the largest eigenvector field of the rate of strain. And this is saying that these optimizers are basically trajectories of this eigenvector field that pass through this point. Actually, they start through this point. So this is going to be one of these curves, gamma, that we were looking for, that are the most attracting curves for a very short time. Okay? That's basically what we have. Okay, now let's do an exercise. So this is basically an oceanic flow that is computed or reconstructed from altimetry. You know, it's called basically, I think, quasi-geostrophic flow from real altimetry data. And now I ask you, this flow is slightly time dependent. And I'm going to play the video. And now I ask you, if someone gives you this distribution of streamlines, and I ask you, where are the regions where you're going to have the largest stretching of material? If you put a bunch of ducts or whatever you want, colorant, and forget about diffusion, where are the regions in this streamline field that will stretch the most? Okay? What would you answer?
It's maximum.
Eh?
Maximum eigenvector.
Sorry?
Maximum eigenvector
Of what?
of the
Oh, the rate of strain, you're saying, yeah. But okay, fine, but the eigenvectors is everywhere. Where would you, I mean, the eigenvector is a field everywhere. Now you have to tell me where. Eigenvector at most can tell you in which direction. But where should you go if you have to bet? Where would you say that there is going to be the highest chance of having large stretching?
Between two vortices, like the region.
You can start going with this intuition, and actually I'll tell you that if you go and look at the books, they would tell you, look, you should really go here, because this is where the streamlines change topology. So that's really, it's basically, this is called saddle point of the velocity field. So here the velocity field is 0, and is hyperbolic. That means that there is going to be a direction that stretches, a direction that compresses. Also, this is also incompressible. So it must be that if one direction stretches, the other one compresses. So then your claim would be that these points between vortices, if you think of vortices as closed streamlines, these points are the ones where you should have largest stretching. Now there are several problems with that. This claim is abusing the results that hold for time-independent systems. So if x dot is equal to v of x and not v of x and t, and you wait for t going to infinity, your answer is correct. And in fact, those are the stable and the unstable money for the fixed points. However, as soon as you have a time-dependent system, and you want to ask, even if it's time independent, and you want to ask what happens for t going to 0, these answers are not the right answer. Okay? Third, these points, the fixed points of the velocity field are frame dependent. That means basically that if you do this assessment by an helicopter or by a boat that is moving or the tower station on board, you get 3 different results, and you don't even know how to connect them. Okay? So let's do that. So this is basically what would you get for asymptotic methods, and this is instead what this technique is telling you. This is telling you that you should go to these different places, the green ones. The asymptotic ones should tell you that instead go to these 3 different places. And all these are frame invariant, the green ones, frame independent. So now what I'm gonna do, I computed this at time t, and I'm just gonna play a movie so that you see in the next, I don't know, whatever time scale, I don't remember what time scale, how these things will deform as a test. But you know, this information have been computed just using one snapshot of the velocity field, like the streamline. But then if you want to say, either you say, hey, these are the most attracting, you trust me, or you have to say, hey, show me how they attract, right? So I'm gonna just use a little bit of data in the future to visualize and prove this thing. If you do that, this is what happens. Okay, so basically, this asymptotic saddle point stretches because there was an objective saddle point. This is what we define, objective saddle point. This stretches also because there is an objective saddle point. Here, this guy barely stretches, and in fact, there is not an objective saddle point. Okay, these traps, basically. Here, you can have huge stretching on a region, basically, that is actually moving. There is no saddle point. And actually, this is a very beautiful exercise example, right? So this guy can move, but still induce a significant stretching. While these guys, even if you have basically a time-dependent velocity field, you can try to do this exercise and say, okay, let me actually, at T1, I'm gonna look for the points such that f of x at T1 equal to 0, the x star. And you're gonna get the fixed point at T1. Okay, then you do this at T2, etc. You're gonna look for basically fixed points of the velocity fields. But these points, the problem is that they are basically anchored, they don't move. They're also frame dependent. Instead, you can have significant stretching also in regions that move. Okay, so why should they be still? This is just what we were expecting in steady dynamical system, but it's not true, because now you say, hey, I told you what I want. I want the curve that attract the most. I don't want to impose that they have to be still, right? You can put this in as an additional constraint if that's what you need, but in general, they can move. Okay, so now think about it. If you would do a search using classical methods, yeah, this would have a very strong consequences, because you would be missing regions where most likely things would get attracted. Okay, so is this clear? Okay.
Actually, I still did not understand why there are some reason which are not in between the vortex, but stretching is more. For example.
Yeah, because I mean, the fact that you think you are between the vortex is coming from some very specific observation. But I can have vortices and I can have no stretching region in between. I can construct an example that is like that, and you don't know what happens in the ocean. I'll give you an example. Think about this. So let's say that you are in a channel, and here you go fast, here you go slow. Now think really about putting some tracers here. What happens? So let's say you put them here, put them here, here. In a little bit, they moved. They all move. But they will squeeze this blurb of thing from this configuration, squeezes here, because it's like when you go, you go out, someone is running faster than you go slow, you're clogging the lane, and you're gonna have a bump of density of people near you in the back, right? So there is no vortex here, but I can still have strong accumulation. There is no fixed point. Okay, now think about it. I can, the ocean is very complicated. So a combination of vortices, this phenomena, and many other that can generate very exotic distribution of attraction, etc. But you don't have to think or care about it. Forget a little bit the intuition. Do this, and you're gonna get the answer. So you know that there could be many different combinations of. Maybe they are also very counterintuitive. Okay. Okay. So what we did. So this was basically, actually many years ago now, unfortunately. So then what we did, we said, so there was a group at MIT that the group of Professor Tom Peacock, he basically wrote a big grant on using recent methods to to mathematical methods to help hazard response. And then they said, okay, let's test this with, in real experiments. And this is the field experiments that we did. So we went, Martha's Vineyard is just, in the east coast of Boston, so just outside Boston, a couple of hours, even less, just really by the coast. And so the Woods Hole Institute of Oceanography is basically a very famous institute of oceanography that has a lot of instrumentation in this area, close to Martha's Vineyard. Specifically, okay, they have boats and all these tools, but specifically they have a region on the ocean here where they have a lot of towers here that can measure the ocean surface. With these measurements and some algorithm, they can actually reconstruct the velocity field in this region. So we said, as a first step, let's see if we have a good quality velocity field, if we can predict where things go. Okay? So what we did, we used this basically sensed surface velocity field. We computed these traps. And then we deployed the drifter and mannequins with boats, and we said, okay, do they go to traps, yes or no, how long does it take, and etc., right? Okay, so this is step number one. And step number two, we said, let's do something more challenging. Let's actually now use a model-based velocity, which is more typical of what happens in the Coast Guard, because when there is basically a search and rescue, it's very unlikely that you're gonna get available ocean surface velocity that is measured. Well, it's in the Titan, it's in the middle of nowhere. How do you know? It's actually also at depth, even there is no way you can measure anything. So you really have to rely on model-based velocities, and this is more challenging because, of course, this means basically that there are a lot of more uncertainties. Okay. So this is the team. So this was my first paper with this. Okay, so the traps, so this thing, I did it with George. This also was done with George. This is when I was finishing up my PhD. So George and I did the theory part. Irina was at WUI, the Woods Hole Institute of Oceanography, is very famous, expert in, in oceanography. She had a lot of data on drifters. Anthony did all the high frequency radar measurements. Shane Ross did provide the mannequins with the tracks. Pierre did all the modeling of the ocean modeling, so the PDs for simulating ocean modeling. Art Allen was the was basically the lead responsible of US Coast Guard for search and rescue, and Tom Peacock was leading all the experiments, and he wrote this huge grant. Okay, so that's basically what we did. So for the first trial, they already had basically a very good data set of drifters deployed in this area and the velocity. So we said, okay, the data is already there. Let's use the velocity. Let's use the trajectory of the drifter. Let's see if they really go where you expect. So distribution of drifters, very spread out. This is the streamline. So you think about now the streamline. There is no vortices here. So you say, okay, actually I would expect that everything is just flowing and nothing is accumulating. Instead, 2 hours after this spread out distribution, you can compute these traps. This S1 is exactly this field. So the more negative it is, the more attracting it is. And these regions are exactly this eigenvector of the rate of strain, the red ones. So you can see that there is going to be traps that are the most attracting. The more blue it is, the more attracting it is. Okay, so this is -10, here is -25. This is real data, and this is the motion of the drifters. So they start clustering in this very spread out distribution. Again, there is no sign of this accumulation on the streamline, because this is actually, it's happening something like this here. It's precisely this that is happening here, which is very hard to spot from looking at the velocity, the streamlines. And then after 3 hours, you get even a stronger alignment, because basically this is getting stronger, more homogeneously stronger. This was a localized stronger, maybe it was the one that generated this huge kick, and then this was becoming, a strong attractor more broadly. And this is after 3 hours, okay? And then if you look at the divergence, this is not really driven by divergence. I have a slide at the end, because this is also something that the other people asked yesterday. And you can have traps, accumulation regions also where the divergence is positive or close to 0. Okay, so you cannot really get this from the planar divergence. I have a slide at the end. Okay, so this was interesting, and we said, okay, this is cool. At least it's going in the good direction that traps can predict the motion of floating objects. Remember that traps are just based on fluids, while these floating objects are not really following the fluid particles. But for very short time, they're very close because the correction hopefully is small. If it's not small, there is no way you can sort of predict them unless you tell me what they're doing or what's their properties, etc. Okay, so that's basically, okay, the video, how they basically cluster. Oh, it's just what I showed you now. So then the second time we said, okay, now let's instead use modeled velocity field, which is not, there is no available data on the surface velocity. It's from the model of Pierre Lermoisat at MIT. What we did, we said, okay, this is the situation with the traps. So this is basically the S2 field, S1 field, what I just told you. Eye attraction is in blue. And then we had, I don't remember, 1, 2, 3, 4, 5, 6, 7, we had 8 drifter and 4 mannequins to deploy with the GPS. So Pierre gave me the model of the model velocity. I computed the traps in this region of the ocean. And then we said, let's actually go out with boats and deploy this bunch of drifters at 10, 15 and mannequins and see where they go. The idea was that they should go to these red structures, the traps, right? So this is a drifter, this is a mannequin. And this is basically how they look when they are in water, basically. This guy is basically in the water, this also guy is in the water. Well, after 1 hour, this is basically how they went. So after 1 hour, they already went so quickly to the traps. I mean, of course, there is some variability. This guy is still sitting here. I didn't really move much after 1 hour. I mean, 1 hour of ocean time scale. Ocean time scale, 1 hour is nothing. Still, these guys, they were able to make it to the trap pretty quickly. After 2 hours, even a stronger distribution of these objects to the traps. Okay, this guy is making his way there. Of course, I mean, this is not, there is a lot of, uncertainties and, things that we're not accounting for. But, in 2 hours, we were really able to predict where things were going. And of course, the traps are evolving. So as soon as I have a new batch of velocity, I can predict and update this S1 field, the E2 fields, and I'll recompute the traps. So this was reassuring because also when we have model-based traps, we can predict the motion of floating objects. Now this is something that I don't remember who asked this question yesterday. I just pull up these slides. So what we did here to understand sensitivity to noise. So whoever was in boat that started at A and B, they had both to deploy a drifter and a mannequin. Mannequin and a drifter, mannequin and a drifter. That means basically that these objects, they started at the same position, and then they underwent slightly different dynamical systems because one guy is a drifter, one guy is a mannequin. The magenta one is a fluid particle that integrated with the model. Okay? And the interesting thing that despite they actually separate, even within 1 hour, these are basically already separated, but the separation is basically collapsing onto the traps. It's a very interesting thing that says, these traps are really capturing the robustness of, the noise or the underlying process. So it doesn't really matter what kind of object is floating unless it's basically a whale that, flies in the water. But otherwise, there are basically very little distinction. I just go to the trap, okay? So different trajectory, same trap. So then if you want to understand this, you can actually do a little calculation and say, okay, this is my Ito process. So I'm going to have a deterministic velocity and some noise, some stochastic part where this is the diffusivity, so the structure of the noise. And then what I can compute is the rate of strain tensor of this uncertain process. If you do the calculation, actually you can see that the rate of strain tensor of a Langevin equation is going to be equal to the rate of strain tensor of the deterministic velocity field plus a rate of strain that comes from the noise. Okay, and this disturbance or noise rate of strain is nothing else than the diffusion tensor of your noise. You differentiate it with respect to x1 plus the diffusion tensor differentiated with respect to x2, and you take the symmetric part. If you do the calculation, this is what you get.
is diffusion, right? So
diffusion is not diffusion, it's just to model random walks. There is nothing is diffusing, it's just that there is a stochastic differential equation, yeah.
But how do you define this tensor? Means how will you in general? So you can measure noise level and then from there you get.
No, you just have a differential equation. This is a vector. V plus d times dw is a vector. Now think of this as a vector field.
No, that part is, I'm just thinking about the diffusion, because usually you consider that diffusion is going to be uniform.
We are gonna get there. Let's see what's gonna happen. It's even more cool because okay, yeah, this you can, you know, in experiments you can measure if there is a structure or to this noise, etc. But very good point, Arma. So let's say this is the way you compute it, but typically D is uniform in space. Because if you know the structure of this noise, then you build it in the model rather than leave it in the stochastic part. So this is interesting because it says that if D is just a function of time, eventually, actually it's not even a function of time, usually you say it's constant, but okay, also if it's a function of time, this guy, so the disturbance part or the random part of this rate of strain tensor is 0. So that basically means that the coherent structures, the traps, will be completely unaffected by the noise. And in fact, this is a sort of a very elegant explanation of what you observe with real experiments, right? Okay, so this is cool because it's saying, oh, you know what, unless you have a very particular structured noise, that it would be hard to know, and also I think very likely hard to realize that there is going to be preferential direction in regions of the ocean on the noise, not on the velocity. So if that's the case, basically these are very robust predictors. Instead, now if you go back to the Lyapunov exponent, if I would do a Lagrangian prediction of I start basically at the initial condition, the growth over some particular time t will be proportional to the largest Lyapunov exponent times square root of t. So Lagrangian predictions will immediately start accumulating noise proportionally to the largest Lyapunov exponent, which tells you how much separation can you achieve times square root of t. These guys are remaining completely unaffected. Okay. So this is basically a way to say that these traps are robust to uncertainties. Okay. This is that's the little other consideration that you can have a very beneficial, very big benefits from considering these instantaneous coherent structures compared to trajectory-based one when you want to address this type of problems. And so basically the summary is that you can have fluid-based traps, basically are short-term attractors of general floating objects. They are 1D curves, so they're really optimal to search. They are robust to uncertainties, very quick to compute. You don't have to compute trajectories, etc. You just have you can actually build them into the ocean models that are available, so that the coaster doesn't have to do anything, just computes, and they have basically these maps of traps. Actually, we are developing a pipeline or a website in which you basically plug in your coordinates, and it's going to tell you immediately where these traps are, depending on if you want to do oil spill, plastic collection, etc. And they are actually hidden to velocity field plots. So this is to say that you can look at vortices, etc. But I mean, these considerations were not really designed. When you look at vortices, etc., nobody is telling you where do things go. Okay, so when you have basically vertical flows, etc. Instead, here the reason it works is that because we design it to be that way. So we said, okay, we asked where are the curves that are the most attracting in this flow, and then we just did the mathematical exercise. And this is now adopted by the Coast Guard as one of their techniques for doing search and rescue. Another application that we did is basically associated with oil spill. So we also showed that knowing the presence of these traps would have predicted, about a week earlier where to go and bound the oil spill containment, the Gulf of Mexico. This is basically a book with NOAA and other collaborators. Rodrigo Duran is an oceanographer in Miami. And there is also a non-profit company in the Netherlands. It's called the Ocean Cleanup. They're trying to clean up ocean plastics, and they are using traps to basically they go with these interceptors, and they want to basically understand where these traps are depending on ocean velocities, etc. And they're basically using this technique to see where to go and find the regions of highest likelihood of finding these traps. So this is basically the full reference, the team, that was a very beautiful joint effort that was very casual. This really came up as a spontaneous, collaboration. And I just want to point out a couple of things. I have maybe 2 slides, this and another one. The first thing was something that one of you asked, I think Sahil asked about, divergence, attraction, repulsion, and this is basically the same question that the reviewer asked when we were submitting this paper. The formation of a fluid patch, there are 2 contributions. We already saw it yesterday, right? So it's an isotropic one, this could grow, or could shear, preserving the volume. Okay, and the total is just, what you observe is a combination of the 2. Now you can have basically attraction in a region of positive divergence, because basically, look at this, basically this guy is going to increase volume overall, but he's going to squeeze in this direction way more than stretching in this other direction. So although the phase space volume is increasing, I can still perceive attraction in this direction. Okay, so basically the divergence is not the only thing you have to look at, unless you are looking at asymptotic attractors in the classical sense of dynamical system, where you wait for infinite time and you really shrink phase space. Okay, but this is basically short time. So even finite time, this is important. In finite time is the same. Attraction in a region of 0 divergence, that's very standard. I attract here, repel here, but the area of the patch is the same of the area of the deformed ellipsoid. And I can also have actually repulsion in a region of negative divergence for the same reason. So basically I can start with this blob, I can shrink its area, the elongated object, but then I can basically have that the repulsion or the stretching is much higher than the compression. So basically this is something that can make sense of something that could be maybe possibly counterintuitive. Okay, so the divergence is not going to tell you necessarily where there is attraction or repulsion. Okay, so this is one. The other thing I decided to put in the slides is something that I think some of you are also already facing, which is that whenever you have data on a finite size data set, so let's say that this was the data on south of Martha's Vineyard, where I had the high frequency radar velocity, or let's say someone does an experiment. This is what you have. The data is you have data, the velocity field data inside this domain, because outside there is no microscope. Now, one thing that you have to be careful is that if the velocity field is outward flowing, means that it's going out. Okay. Then there is a problem, because you start here, and then by a certain time, this trajectory has left the domain. So from that point on, there is no way you can tell me what Lagrangian process has happened here, because you don't have more information. Okay, and if you, in this particular oceanic example, you can see basically that the more you push the integration time, the least information you have available, because most of the trajectory are likely leaving your domain. Okay, so this is an important thing when you have real data. There is usually a finite size domain in which you have data, and if you want to start cranking the integration time, you're gonna not be able to address things. But the Eulerian methods are always there, because you have velocity here, you're gonna get coverage forever. This is also something that you have to consider. And in fact, if you remember the slides on the chick, there were basically regions that became white progressively. A larger white region on this domain is because these cells were actually leaving. So I didn't know how to. The repeller number 1 was actually very eroded, from the experimental data for this reason. Okay, because cells are leaving, so unfortunately you cannot really address this point. So another thing you can do is define the notion of instantaneous Lyapunov exponent. So if you, instead of expanding Cauchy-Green, you expand the finite time Lyapunov exponent, which I'm doing here, you actually recover the largest eigenvalue of the rate of strain. So the instantaneous repellers, you can read them from the largest eigenvalue of the rate of strain. Okay, a diagnostic to understand short time attractors, you can just look at the smallest eigenvalue of the rate of strain, which is S1. If you just don't want to be so precise and tell you in which direction I want to pretend normal, attraction. So if you're not caring about enforcing normal attraction, but you can also accept, shear type attraction. Okay, then you can just look at S1 instead of basically looking at also the eigenvectors. And so now you can have basically short time attractors and short time repellers. These are Eulerian, so you can also start doing optimal control. So because controlling Lagrangian attractors and repellers is very complicated, because basically you have the velocity field that you don't know, then there are functional of the velocity, which is the trajectory, and then the Lyapunov exponent, a very nonlinear function of the trajectory. So doing optimal control for attractors and repellers, for instance, could be, I don't know, how should I design the distribution of myosin such that I create an attractor? Okay, that's very hard to do mathematically, but then you can start, at least we started asking, can I create a short time attractor? And the answer is yes, we can, and you can actually use optimal control and active nematics, in which, in this case, we use the same equation for the chick development that I showed in the morning, and I said now myosin is not a field that evolves by itself, but is my control variable, and I asked, how can I create the desired distribution of short-time attractors or repellers tuning myosin? Because, generalizing a little bit development, eventually development can be thought as, a set of attractors and repellers, in which basically the attractors make sure that the right cells go to the right place, and repellers make sure that distinct cells, they separate from each other. So eventually you say, okay, maybe the embryo is trying to optimize attractors and repeller geometry, and then, this is a first attempt in which basically we controlled M to achieve a particular configuration attractors and short-time attractors and repellers. And I think that's all I have. And if you have more questions, or if you have questions, I would be very happy to take them. Yeah.
Thanks.
Question?
Yeah.
Actually, I just want to understand in the ocean model.
Yeah.
So where there is a short time attractor.
Yeah.
So since we know that the attractor won't be constant for longer time, they are varying very short in time.
Yeah.
So how one can track it within that short time, because lots of motion is going on.
Yeah. No. You can. What do you mean? You can track it. This is, for instance, so this is real, this forget models, this is real data. So you see this attractor pops up and then I track it for, 1 hour to the next hour. These are all structurally stable features. They are very robust. So they will not disappear in a split second. They can deform a little bit, move a little bit, but they will not disappear. Okay? So they're always there. Then you can also ask, how strong is this attractor? What's their robustness? You can say if I perturb time a little bit with Poincare section arguments, you can actually, or implicit function theorem, actually you can understand, or actually classify all the present attractors and say short-time attractors and say this guy is going to be most robust than the other guy. So this could be also an additional information you can use to say, okay, I'm going to chase this guy instead of the other guy. Yeah, now, for instance, an interesting question could be, similar to what other people asked yesterday. You say, okay, I already know the equations that govern ocean surface dynamic, and I ask you, what forces generate these objects? This is also something we're trying to investigate, right? Because there are a lot of forces on the force balance on Navier-Stokes, but then can you actually say that, contribution of a couple of forces when they are in a particular alignment with each other guarantee the presence of attractors? So you can start asking these type of questions on the cause of this, and then maybe say, okay, can I design a flow that has these particular attractors? Yeah. Yeah, other questions? Okay.
So just to get it, so suppose if we have an Eulerian field, you can calculate the strain-rate tensor. From there, you calculate the eigenvalues and eigenvector for what is the minima of S1, and where the largest eigenvalue is intersecting, and that is going to be the point where attract.
Yeah, that's it. So that's it. Yeah.
That's what you have.
Yeah, actually, guys, in this paper, on my website, in the publication tab, there is a code. That is, there is a code for computing traps. So you can basically use it. You plug in the velocity field you want, and it's already there. But yeah, that's the result. Yeah. 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 Yeah.
And usually, so of course it is going to be dependent on like if the dynamics is really fast, so attractor will be moving around in this.
Yeah, we're moving around. Yeah. Yeah.
Do a like correlation and try to see the lifetime of these structures.
Yeah. I mean, the ocean surface dynamic here, it is fast.
Yeah.
Actually, especially here because there is a channel. So this is very fast because usually when you go, what I understand is that basically whenever you are near coastal region, the strength of the ocean currents are much faster because there's boundary layers, some of this kind of stuff. But in the middle of the ocean, it's way more calm. And these experiments were done close to the coast because it's easier to reach. Yeah. Okay.
Okay. Thanks.
Of course. Yeah.
So in the second half, what will retain from where we left? So what we do first try where we left, because unless you figure that out, we cannot move forward. Okay? So the aim is that spend some time, try to use your Rollman flow again, use the data, and try to see if you can find the finite time of the exponent. That you should definitely try to do. And once you manage to do that, then we will. Then you have to write down the equation for the inertial heavy particles and try to see whether they are going to get accumulated in the strain dominated region or not. And if that happens, then you have to check what is happening with FTL. Okay, that is one thing. And then the third aspect would be like, so actually first we wanted to give you real experimental data, but data is very noisy and it will be. We realize that it is too challenging task to handle. So it's better you do something which is much more smooth and much more controllable.
So it's exactly the same. Once you have the code for a velocity field, then you can just use it for any other code.
Yes. Yeah. It's better to handle the flows that we understand very well. So so for tomorrow, what you will be doing, you will be taking the same velocity field that you have already done. You have looked at the vorticity field, right? So the flow. You take that flow and try to calculate the short time Lyapunov exponents. Like use the Eulerian field, you find where is the minima of S1 and which the eigenvector corresponding to largest eigenvalue where it is going to intersect, and that's going to be your attractor.
Yeah, okay, you can also guys. I mean, it's good to plot the eigenvector field.
Important. Plot, visualize, get a feel for it first.
Yeah.
Then you
don't compute the trajectories of the eigenvector field because that's a little bit, a delicate thing. Or you can go to my website and then use the code that is already there. But even if you just plot the S1, it's already a good.
That will say what is the point. And then
exactly.
You need to find the direction. So
So
that's the plan. So today you will still be dealing with the agent-based Lagrangian data. We expect that you get a good control on FTL finite time Lyapunov experiment, and then we will not be covering that tomorrow. So tomorrow it's a new problem. Okay. So we hope that you manage to do it. And please, if you're not able to fix it, note down your questions. We'll be around in of course PS210, and tomorrow also will be available. And for Eulerian, that's okay.
yeah.
Okay. Okay, thank you everyone. So we'll disperse for lunch and then we'll meet at 2:30 second floor. Thank you.
Okay. So today, frutta, non pa?
Sorry?
Fruit.
Yes. Let's go and grab. I think this will also work out. It has been a long session for you.
Yeah. Yeah, I don't know why, but basically, even when I teach